In this video we will talk about the oxygen dissociation curve and what does this curve signify and tell us about the affinities of hemoglobin and myoglobin towards oxygen. So this curve is a graph in which the y-axis shows the percentage saturation. Percentage saturation with oxygen, like how much it is getting, how much hemoglobin or myoglobin is getting saturated with oxygen. That means how much it is activated and at what partial pressure. So this is the partial pressure of oxygen. So y-axis is the saturation, percentage saturation with oxygen. This is the partial pressure of oxygen. In case of hemoglobin, its affinity towards oxygen is low than that of myoglobin. So it has somewhat sigmoidal curve in nature. This is sigmoidal in nature. So this is the curve for hemoglobin, because hemoglobin has a lower affinity towards oxygen. That means, in other words, we can say that hemoglobin can deliver oxygen more easily to the tissues than that of myoglobin, and that is what we need. Okay, and in case of myoglobin, this is hyperbolic in nature. Okay, so this is hyperbolic in nature. One more thing about myoglobin is that myoglobin in the skeletal muscle, hemoglobin, which is bound to oxygen, it transports oxygen to the myoglobin and myoglobin stores oxygen in it. When muscular activities is high, at that time this oxygen is required to provide energy to the muscles. Now, when this oxygen is depleted or exhausted in the muscles, at that time muscles they respire anaerobically, and that produces lactic acid. Remember, we did in glycolysis chapter, anaerobic respiration give rise to lactic acid, not pyruvic acid. So on depletion of oxygen, myoglobin respires anaerobically and produces lactic acid, and this lactic acid is picked up by the liver from the blood, and that is converted into pyruvate, which can enter into the in other reactions, in other oxidative reactions. So this is about the myoglobin. So this is the curve for myoglobin, and this is hyperbolic in nature. This is myoglobin. This is hemoglobin. So you see, suppose if I am taking 50% saturation, 50% saturation, the partial pressure of myoglobin is so low. This is the partial pressure of myoglobin, and this is suppose 1 millimeter of mercury. Okay, and at the same saturation, you can see the partial pressure of hemoglobin is so high, around 26 millimeter of mercury. So from here it is clear that hemoglobin has lower affinity towards oxygen. That's why it requires very higher partial pressure to get half saturated. So more the affinity of the hemoglobin or any substance towards oxygen, lesser will be its partial pressure. So more affinity for oxygen, it will lower will be the partial pressure of oxygen. You can see for myoglobin the partial pressure is so low. That means when it is becoming 50% saturated, it just requires 1 millimeter of mercury, so much affinity towards oxygen. So that means this will bind tightly to the oxygen. It will bind tightly, its bond will be very strong. Clear? So myoglobin binds to that oxygen with one hemidine. So it has higher affinity. That means hemoglobin can deliver oxygen more easily, and this delivering of oxygen to the tissues, this is the steep. You can see the rise here is steepest. That means it can deliver oxygen more easily to the tissues, so it's rising. Now, suppose pH, so what is happening actually? So this is the cell which is respiring, okay? So this is producing carbon dioxide. So this is producing carbon dioxide coming into the plasma. Now this carbon dioxide joins with H2O and forming carbonic acid, and this is unstable and breaks up into H plus and bicarbonate. This is formed by carbonic anhydrous. This is forming this proton and bicarbonate. So you see, proton's concentration is increasing in the cells, in the tissues. So acidity is increasing. Now more the acidity or more the lesser the pH. So lesser the pH, that means more is the acidity. That means more acidic. This decreases the affinity of hemoglobin towards oxygen. So this decreases affinity towards oxygen, affinity of hemoglobin. So this is Bohr's effect. So Bohr's effect is where when H plus ions are increasing, when protons are increasing, pH is decreasing, making the environment more acidic, hemoglobin's affinity towards oxygen is decreasing. That means it is delivering oxygen to the tissues and it is combining with this H plus, maintaining the pH level. So this hemoglobin can act as an buffer. So it is maintaining the pH of the blood. Okay. So this is what is your Bohr's effect. This can also happen when carbon dioxide concentration is also high. Same thing, because if carbon dioxide concentration is high, it will give more H plus. So this is what is Bohr's effect. So this is happening in the cell. Actually this is happening, this whole thing is happening in this cell. So in the this H plus and bicarbonate is formed. This H plus is making the cell acidic. Hemoglobin is coming. Its affinity has been decreased, giving out the oxygen. So it is delivering the oxygen combining with this H plus. So maintaining the pH. So acting as the buffer. And what happens to this? Now we have sodium ions. This comes out and joins with this sodium ion and forms sodium bicarbonate. So this negative ion is coming out. Negative charge is losing from the cell. So to maintain this balance, chloride ion is entering into the cell. Chloride ion is entering into the cell, and this effect is called Hamburger's effect or chloride effect. So what is happening? When the cell is respiring, it is giving out carbon dioxide, which is combining with oxygen, water that is present in the cell and forming carbonic acid. that splits up into hydrogen ion and bicarbonate ion. Now the hydrogen ion that is formed, it is lowering the pH, making the environment acidic. Hemoglobin's affinity towards oxygen is decreasing in this acidic environment, and thus it is delivering more oxygen in the tissue. This is Bohr's effect. On the other hand, so hemoglobin is taking out that H+, so maintaining the balance in the cell, that is acting as the buffer. On the other hand, the bicarbonate that is formed, that is coming out of the cell and joining with the sodium ion, forming sodium bicarbonate. But the cell is losing negatively charged. So to maintain that balance, chloride ions are coming inside the cell. So to balance the loss of negative charge, and this is Hamburger's effect. Now, what is the effect of this Bohr's like pH or lowering pH on the oxygen dissociation curve? In this case, the curve of hemoglobin in the oxygen dissociation curve, it will shift to the right. So this will shift to the right. Let me draw with the red pen. So it is shifting to the right. So from here, it has come here. That means its affinity is further decreasing when the pH is low. That means when hydrogen ion concentration is high. So it will shift to the right. Thus, it is implying that its oxygen affinity is decreasing. So it is delivering more oxygen towards the tissue. So this is the effect of Bohr's on the oxygen dissociation curve. It shifts the curve towards the right in presence of Bohr's effect. Now there is another thing, that is another compound is there which is called 2,3-bisphosphoglycerate. This is we have another compound, that is 2,3-BPG. This 2,3-BPG is formed from 1,3-BPG. In glycolysis, we have seen glucose undergoing so many reactions, and finally it is forming 1,3-bisphosphoglycerate. This is one intermediate that is formed. This forms 3-phosphoglycerate. This we have discussed that this is a substrate level phosphorylation. This 1,3-bisphosphoglycerate can undergo reaction and it can form 2,3-bisphosphoglycerate. Okay, so this can also form 2,3-phosphoglycerate by the enzyme phosphoglycerate mutase. This can again give rise to this 3-phosphoglycerate by 2,3-bisphosphoglycerate phosphatase. Okay, so this 2,3-bisphosphoglycerate has effect on this oxygen dissociation curve. How? It binds to hemoglobin. It binds to hemoglobin, thus lowering the affinity of hemoglobin towards oxygen again. So, we have seen that there are chains, alpha chain, beta chains, and inside you have hemoglobin. And you can see there is a formation of this pocket that is formed. So, inside this pocket, this 2,3-BPG comes and binds to this or stays here. Okay, and the negatively charged phosphate group forms a kind of bond with the positively charged amino acids which are present in the globin chain. So this is how this 2,3-BPG binds to the hemoglobin. So this hemoglobin has a higher affinity towards this than that of oxygen. So when this 2,3 BPG is bound to this in this pocket, when it is sitting here in the pocket, it decreases the oxygen affinity. Now this heme cannot bind to oxygen. Okay? So this decreasing the oxygen affinity, so the oxygen can be delivered easily to the tissues. And when this is not present, oxygen can bind to it. So this again shift the curve towards the right. So this is again due to 2,3 BPG. So this shifting of the curve, this oxygen dissociation curve towards the right can be due to two reasons. One is due to the Borscht effect. When the pH of the environment is decreasing, that is, it is becoming acidic, at that time the affinity of hemoglobin towards oxygen is also decreasing. So it is shifting the curve towards the right. At the same time, this 2,3 BPG that is present, or when it is elevated, it binds to hemoglobin. Thus, it is also reducing the affinity towards oxygen. Thus, again shifting the curve towards the right. So, this part is very, very important in your exam point of view. And there is one more thing, when the elevated BPG level is very high, you have this condition of breathing problem, like COPD is seen in those patients in where 2,3 BPG level has increased, because when this level has increased, so it is binding to hemoglobin more. So hemoglobin is not available for oxygen to bind. So those patients cannot breathe easily. They have breathing troubles in that. Okay. So this is your overall oxygen dissociation curve. One more thing is that the hemoglobin that is present inside our body, that is adult hemoglobin. So the 90% of our hemoglobin is HbA. Okay. So this is formed of alpha 2, beta 2 chains, and mainly it is 90% of the hemoglobin is made up of HbA. Now there is fetus hemoglobin which is present in the newborns or in the fetus, and this HbF has a very high affinity, oxygen affinity, unlike this one. Okay. So this fetus hemoglobin, it is made up of alpha 2 but gamma 2 chain. Remember this. Clear? It has very high oxygen affinity, so the maternal oxygen can be easily transferred to the fetus because of its high affinity towards oxygen, and it has very low affinity towards the binding with 2,3 BPG. Thus, it can bind to oxygen more tightly. Okay, but if you strip off this 2,3 BPG from both HbA and HbF, they both have the same affinity towards oxygen. Now, in embryonic hemoglobin is mainly made up of this alpha-like chain, which is zeta 2, and beta-like epsilon 2 chain. This is synthesized by this embryonic yolk sac. So they have zeta 2 epsilon 2 chain in the embryonic hemoglobin, but in the 5th month of gestation they are shifted to the liver and then to the bone marrow. In the bone marrow, where this is changed to fetal hemoglobin. Okay. Next we have another form of hemoglobin which is A2. This is made up of alpha 2 delta 2 chain. And this is present just before the birth. After birth, you do not have this one. Okay. Another one is HbA1c. This is actually glycated hemoglobin. That means one hexose bond or any glucose residue. Here, glucose residue is bound to N terminal of valine in the beta chain. So the beta chain has valine group, and in the valine group, in the N terminal of the valine group, glucose gets attached. So this is glycated hemoglobin, and mainly this kind of hemoglobin is mainly present in those patients which have diabetes mellitus. So these are the different forms of hemoglobin that you should know about. Okay. So this is all about your exchange of gases.